This video is about cell organelles and cell division. Cell is the basic unit of all living organisms. Coming to the cell classification, the organisms can be divided into prokaryotes and eukaryotes. Prokaryotes are single cellular like archaea and bacteria. In eukaryotes, there are two further classification like single cellular and multicellular. Single cellular are protists and multicellular are fungi, plants and animals. There are 6 major kingdoms in the basic classification of organisms: eubacteria, archaebacteria, protista, fungi, plantae and animalia. There are different systems of classification of organisms. 5 kingdom system consists of Monera, Protista, Plantae, fungi and animalia. 6 kingdom system has eubacteria, archaebacteria, protista, plantae, fungi, animals. In the 3 domain system, eubacteria, archaebacteria and eukaryota. In the 8 kingdom system, eubacteria, archaebacteria, archaezoa, chromista, protista, plantae, fungi and animalia. 5 kingdom system was most used. Kingdom Archaebacteria is prokaryotes. Here no true nucleus, no membrane bound organelles like mitochondria or chloroplasts. No peptidoglycans in the cell wall, reproduce asexually by budding and fusion. The size is very small like 1 to 10 micrometer. Extreme environment are adaptable here. In eukaryotes, they have 2 nucleus, membrane bound organelles. Cell size is generally 10 to 100 micrometer. At least 4 lineages are observed like protista, plantae, fungi and animalia. This is how we can differentiate the prokaryotic and eukaryotic cell. In prokaryotes, the nucleus is not distinct. It is in the form of nuclear zone, that is nucleoid. Nuclear membrane is absent. In eukaryotes, nucleus distinct with well-formed nuclear membrane. Prokaryotic is single membraned cell bodies like mesosomes present, endoplasmic reticulum, plastids, mitochondria, microbodies like lysosomes and Golgi body absent. Whereas in eukaryotes, double membrane cell organelles like chloroplast, mitochondria, nucleus, single membrane like Golgi apparatus, lysosomes, vacuoles and endoplasmic reticulum. There are half membraned structures also like glycosomes, peroxisomes and all. The ribosomes of prokaryotes are 70S type and eukaryotes 80S. In eukaryotes also, mitochondria and chloroplasts are 70S type of ribosomes. In prokaryotes, no compartments. In eukaryotes, distinct compartments in the cell like cytoplasm and the nucleus. In prokaryotes, only 1 chromosome per cell in circular form. In eukaryotes, it is depending upon species. Number of chromosomes per nucleus varies from 2 to many. And here chromosomes are in linear form except in mitochondria and chloroplast. In prokaryotes, the chromosome has single double-stranded circular DNA molecule and is not associated with histones, whereas in eukaryotes, the complexity of chromosome is quite high. In prokaryotes, chromosomes lack centromere. In eukaryotes, each chromosome has 1 centromere that divides the chromosome into 2 arms. However, if the centromere is terminal, the chromosome would have only 1 arm. Difference between plant cell and animal cell. The plant cell has cellulose cell wall present external to cell membrane. In animals, no cell wall. Outermost structure is cell membrane or plasma membrane. In plant cells, vacuoles are usually large, but in animals, vacuoles are generally absent, or if present, they are usually small. In plant cells, Plastids are present, in animals absent. Plants Golgi body present in the form of units known as dictyosome. In animals, Golgi body is well developed having two sister names. In plants, centrioles absent, and in animals, centrioles are present. Here there is a pictorial representation of plant cell and animal cell. Plant cell is in square form and animal is in ovular form. Coming to plant cell, the special features are they have cell wall, that is protection and support membrane, then chloroplast is for photosynthesis, and large central vacuoles for storage and increasing the surface area. They give stiffness to plant cell also. Different organelles are the factory components with function are given here. For support, cell wall, cell membrane, cytoskeleton, microtubules are involved. For control, material entering and leaving are cell membrane, pores. Internal transport system is endoplasmic reticulum, powerhouse, mitochondria. Control centers, nucleus, organelle DNA for mitochondria and chloroplast. Production of key products, ribosomes, endoplasmic reticulum and chloroplast. Packaging center for shipment of products, Golgi apparatus and endoplasmic reticulum. Shipment of materials out of cell are Golgi apparatus and vesicles. Storage of liquids and solids, vacuoles, vesicles, plastids. Recycling centers are lysosomes and peroxisomes. Converting light energy to chemical energy are chloroplasts. Allow new cell factories to the produced like nuclear DNA, centrioles and cell wall. Coming to individual organelle, the first and foremost is cell wall that found outside the cell membrane. Remember, here I am discussing about the plant cell and the division. Cell wall is non-living layer. It supports and protects a cell found in plants, fungi and bacteria. It is characteristic feature of plant cell consists of cellulose, macrofibrils embedded in a matrix. The matrix is usually composed of polysaccharides like pectins or lignin. Functions to provide support in herbaceous plant, provide mechanical strength. The strength may be increased by the presence of lignin in the matrix between cellulose fibers. It permits movement of water through the plant, in particular in the cortex of root. Cell wall develop a coating of wax cutin, the cuticle, to decrease water loss and risk of infection. It gives a shape of the cell. Sometimes cell walls are modified to act as food reserve, example hemicellulose in some seeds. Cell walls possess minute pores through which plasmodesmata can pass for leaving connections between cells and allow all the protoplasts to be linked in a system called symplast. The second organelle is plasma membrane. Plasma membrane is composed of double layer of phospholipids and proteins. This surrounds outside of all cells, controls what enters or leaves the cell. Then leaving layer. In the previous slide I forgot to mention the typical characters of parts of cell wall, that is primary middle lamella, primary cell wall and secondary cell wall. Middle lamella is most important, which connects the interlinked cells, and adjacent to that primary cell wall is observed, and just adjacent to primary cell wall secondary cell wall functions. Cell wall is non-living material, whereas plasma membrane is living layer. The type of cell which contains cell membrane is both plants and animals. The location is outer boundary of cell, surrounds the cytoplasm, separates the cell from its environment. It is made up of a double layer of fats called lipids with some proteins scattered throughout. The proteins form tiny openings or passageways in the membrane called pores. The function of cell membrane is to control what moves into and what out of the cell. Only certain materials can get into or out. Example, food, oxygen and materials go in and wastes and cell products move out. It provides protection and support for the cell. It is flexible so it can change shape under pressure. By allowing things to move in and out, it helps to maintain homeostasis. Cell membranes are described as selectively permeable since, apart from small molecules such as water, large molecules like glucose, amino acids, glycerol and ions can diffuse slowly through them, and they also exert a measure of active control over what substances they allow through. As organic solvent like alcohol penetrate membranes even more rapidly than water, this suggested that membranes have non-polar portions. In other words, they contain lipids. After careful chemical analysis, it is found that membranes are comprised almost entirely of proteins and lipids like phospholipids, glycolipids and sterols. The fluid mosaic model can be used to describe the detailed structure of plasma membrane. Coming to fluid mosaic model, that is most accepted model showing plasma membrane structure. It was put forward in the early 1970s by S.J. Singer and G.L. Nicholson. The protein molecules vary in size and have a much less regular arrangement. Some proteins occur on the surface of the phospholipid layer, while others extend into it. Some even extend completely across it. This is viewed from the surface. The proteins are dotted throughout the phospholipid bilayer in a mosaic arrangement. The hydrophilic phosphate heads of the phospholipids face outwards into aqueous environment inside and outside the cell. The hydrocarbon tails face inward and create a hydrophobic interaction. Hydrophilic molecules will be repelled by the hydrophobic tails of the phospholipids. They can pass the membrane only through the pores formed by proteins that spans the membrane or by protein carriers. The phospholipids are fluid and move about rapidly by diffusion in their own layers. Membranes also contain cholesterol, which disturb the close package of phospholipids and keep them more fluid. This can be important for organisms living at low temperatures when membranes can solidify. Cholesterol also increase flexibility and stability of membranes. Without it, membranes break up. Glycoproteins and glycolipids are important recognition features of the cell. Cytoplasm. Cytoplasm is water plus chemicals. The function is to hold the organelles in position. Cytoplasm is all living contents of a cell within the plasma membrane, exclude nucleus and large vacuoles. That means protoplasm is cytoplasm plus nucleus, whereas protoplast is protoplasm plus plasma membrane. The cytoplasm is present both in plants and animals, or we can say protoplasm. Location is found inside of the cell membrane but outside the nucleus. Protoplasm is clear, thick, jelly-like material, contains large amount of water, about 70%, and chemicals. It may sometimes appear to be grainy. This comes from the organelle floating in it. The function is they contain all the organelles outside of the nucleus, and cytoplasm is constantly moving or streaming through the cell. Cytoskeleton. The location is throughout the cytoplasm. A web of long tubes and fibers are made up of protein. Function is that support the cell and helps the cell to keep its shape, also helps to contain cells to move. Endoplasmic reticulum. It's a complex network of double membrane extending throughout the cytoplasm of all eukaryotic cell. It is an extension of the outer nuclear membrane. Types of endoplasmic reticulum are rough and smooth. Rough means endoplasmic reticulum lined with ribosomes used for transporting the proteins synthesized from the ribosomes. Smooth endoplasmic reticulum means lacking ribosomes and concerned with the synthesis and transport of lipids. Functions: biosynthesis. The smooth endoplasmic reticulum may assemble fats, steroids and carbohydrates, and rough endoplasmic reticulum produce proteins, especially enzymes. Transportation. It's a complex network of passageways that extends throughout the cytoplasmic fluid. Therefore, wastes and nutrients are transported intracellularly. For support, it forms a sort of cytoskeleton that to contain the shape of the cell. Increased surface area of the cell that has network like endoplasmic reticulum provides lot of surface area for the biochemical reactions to occur. Storage. In striated muscle cells, there are a highly specialized smooth endoplasmic reticulum called sarcoplasmic reticulum, which stores calcium ions and is involved in the muscle contraction. Detoxification. In the liver cells, both rough and smooth endoplasmic reticulum are involved in detoxification of various drugs. Nucleus. It stores the genetic material of the cell in the form of multiple linear chromosomes, surrounded by a nuclear envelope composed of two phospholipid bilayers. In chromosomes, DNA is organized with proteins to form chromatin. It is found in all eukaryotic cells except in mature phloem sieve tubes elements and the mature red blood cells of mammals. The shape of the nucleus is sometimes related to that of the cell, but it may be completely irregular. Almost all cells are mononucleate, but binucleate cells, some liver and cartilage cells, and polynucleate like white blood cells also exist. It is bound by a double membrane, that is nuclear envelope. The envelope possesses many large pores which permit the passage of large molecules like RNA. The cytoplasm-like material within the nucleus is called nucleoplasm. It contains chromatin, which is made up of coil of DNA bound to proteins. The chromatin are genetic materials of the cell. In a resting cell or interphase, it appears as a network of tiny granules. During cell division, the chromatin granules are reorganized into filaments called chromosomes. Functions: it has genetic material like chromatin, acts as control center for the activity of cell. Nuclear DNA carries the instructions for the synthesis of proteins. It is involved in the production of ribosomes and RNA, essential for cell division. Location: it is found within the cytoplasm, separated from the cytoplasm by nuclear membrane. It has the largest organelle made up of three parts. One is nuclear membrane, chromosomes, and nucleolus. Nuclear membrane. It's a thin layer surrounding the nucleus, contains pores to let materials in and out. Chromosomes. Thin thread-like structures made up of DNA that is controlling cell activities and providing the information or receive the ribosomes need to make proteins. Nucleolus that makes ribosomes which make protein. Function of nucleus is to control all the cell activities, control which proteins are made. Chromosomes contain genes that control the characteristics of an organism and pass on the traits. Nucleolus. It is the most important part in nucleus, appears as a rounded, darkly stained structure inside the nucleus. It is one or more nucleoli may be present in the cell. The change intensely because of the large amount of DNA and RNA it contains. During nuclear division, nucleoli seem to disappear, but this is because the DNA disappears. They reassemble after nuclear division. Function is making ribosomes. Ribosomes. A small 20 nm in diameter and non-membranous structure, contains two subunits. One is large 70S and small 80S. It is present in large numbers in both prokaryotes and eukaryotes, may occur in groups called polysomes, and may be associated with endoplasmic reticulum to form rough endoplasmic reticulum or occur freely within the cytoplasm. It is made up of roughly equal amount of ribosomal ribonucleic acids, that is RNA, and proteins. Function: it acts as a binding site for protein synthesis. Golgi bodies. These are stacks of flattened sacs, have a shipping side, that is transphase, and receiving side, that is cisphase. Receive proteins made up of endoplasmic reticulum. Transport vesicles with modified proteins, pinch off the ends. Golgi apparatus is a secretory organelle. It has a similar structure to smooth endoplasmic reticulum but is more compact. It consists of flattened membrane-bound sacs called cisternae together with a system of associated vesicles called Golgi vesicles. In plant cells, number of separate stacks called dictyosomes are found, while in animal cells a single large stack is thought to be more usual. At one end of the stack, new cisternae are constantly being formed by fusion of vesicles, which are probably derived from buds of smooth endoplasmic reticulum. This outer or forming face is convex, whilst the other end is concave inner or maturing phase, where the cisternae break up into vesicles once more. Functions of Golgi complex: packaging, glycosylation, and concentration. Packaging: materials that are manufactured elsewhere in the cell move along the endoplasmic reticulum into Golgi apparatus, where they are packaged and being pushed to the ends of the organelle and pinched off into small bubbles like secretion vesicles. Glycosylation: many cell secretions are in the form of glycoproteins, and although considerably glycosylation takes place in endoplasmic reticulum, the finishing touches in a Golgi function. Concentration: dilute secretion is firstly concentrated in the Golgi apparatus before discharge. Transportation: when digested, lipids are absorbed as fatty acids and glycerol in the small intestine. They are resynthesized to lipids in the smooth endoplasmic reticulum, coated in proteins, and then transported through the Golgi apparatus to plasma membrane, where they leave the cell, mainly to enter the lymphatic system. Lysosome formation, membrane differentiation, making cell wall are the other functions. For membrane differentiation, many membranes are synthesized at the endoplasmic reticulum, transferred to the Golgi apparatus, where modifications occur, and fully modified is then added to the plasma membrane by fusion of Golgi vesicles during exocytosis. Enzyme production: example, digestive enzymes of pancreas. Making cell wall that secretes carbohydrates, those involved in cell wall formation. Lysosomes: the size are similar to mitochondria, being 0.2 to 0.5 micrometer in diameter. Functions of lysosomes are endocytosis, exocytosis, autolysis, and autophagy. Endocytosis means process whereby extracellular materials are brought into cell and then included inside the lysosome for digestion. Exocytosis is the release of their enzymes outside the cell in order to break down other cells. Autolysis is self-destruction of cell by release of the contents of lysosomes within the cell. Autophagy is special type of controlled autolysis in which constituents are selectively degraded by inclusion within a specialized lysosome. Microbodies. These are membrane-bound vesicles, contain enzymes not part of the endomembrane system. Glycosomes in plants contain enzymes for converting fats to carbohydrates. Peroxisomes contain oxidative enzymes and catalase. Coming to mitochondria. This is surrounded by a double membrane, has its own DNA, folded inner membrane called cristae that increase surface area for more chemical reaction. Interior is called matrix. Mitochondria are spherical or rod-shaped, scattered throughout the cytoplasm of all eukaryotic cells. The double membrane bounded, the outer of which controls the entry and exit of chemicals, and the inner membrane is folded inwards, giving rise to cristae as to increase the surface area on which respiratory processes take place. Stacked elementary particles are special bodies or spherical bodies located on the inner membrane. They contain enzymes for phosphorylation of ADP, that is ATP formation process. The remainder of the mitochondrion is the matrix. It is a semi-rigid material containing protein, includes all the soluble enzymes of Krebs cycle and those involved in the oxidation of fatty acids, lipids, and traces of DNA. Functions of mitochondria. First and foremost is energy metabolism and respiration. Major function is to produce ATP from foodstuffs via Krebs cycle and respiratory chain. These are the sites of terminal catabolism of foods, that is the primary degradation of those compounds occur in the cytoplasm. Heat production. Energy of oxidation is dissipated as heat instead of being converted into ATP. So, mitochondria is the site of to produce heat to maintain body temperature. Amine metabolism. Some amines are metabolized in mitochondria. Vacuoles. These are membrane-bound structures with various functions depending on the cell type. There are different types of vacuoles like central vacuoles, contractile vacuoles in some protists, and central vacuoles in plants. Vacuoles are used for storage. Vacuoles are fluid-filled sac bounded by a single membrane called tonoplast in plant cell. It contains a soluble of mineral salts, sugars, amino acids, wastes, and sometimes pigments. These substances are collectively called cell sap. Animal cells contain small vacuoles, but plant cells have large central vacuole. The functions are support and growth, storage of pigments, and as used as lysosomes, and temporary store for waste and food. Supporting and cell growth. Water enters the concentrated cell sap by osmosis so turgor pressure builds up within the cell. Osmotic uptake of water is also important in cell expansion during cell growth. Storing pigments. Sometimes contain pigments that is responsible for the colors in flower, fruit, buds, and leaves. This is important in attracting insects, birds, and other animals for pollination and seed dispersal. As lysosomes, sometimes it may contain hydrolytic enzymes. After cell death, the tonoplast loses its differential permeability and the enzyme escape, causing autolysis. Temporary stores for waste and food. Chloroplast. Organelle present in cells of plants and some other eukaryotes, contain chlorophyll for photosynthesis, surrounded by two membranes. Two main parts are grana and thylakoids. Grana and stoma are the grana is a particle and grana is stacks of thylakoids. It is the most common plastid in the plant cell, bound by double membrane envelope. Stoma is a homogeneous matrix which contain enzymes for carbon dioxide fixation processed, that is dark reaction in photosynthesis. Grana are scattered within the stoma. Each granum consists of membrane bound disc shaped vesicle or thylakoids arranged like a pile of coins. The grana connected with each other by intergranal lamella. Both are the sites for light reaction of photosynthesis. The thylakoids are formed by double layers of thylakoid membrane. Contains all the energy generating system like chlorophyll, electron transport chain and ATP synthase. Small amount of DNA is present within the stoma. This suggests that the chloroplasts are also kind of semi-autonomous organelles. Chloroplast might be residue of primitive algae once lived symbiotically in the cell of non-green organelles. That's why the DNA material present in the chloroplast are explained with the help of endosymbiotic theory. Functions are photosynthesis, synthesis of fatty acids. In stoma, carbohydrates are converted into fatty acids, that is of ATP and NADPH. Reduction of nitrate to ammonia. Peroxisomes. These are most important aspect of cell structure, which is functioning as fatty acid metabolism. They are hence different from lysosomes in that they are involved in metabolism rather than in maintenance of the cell. Remember, peroxisomes are involved in metabolism, not in cell maintenance. They are assembled from different proteins synthesized in the cell by ribosomes. The term peroxisome is per means peroxides, oxy means oxidation. So peroxisomes are those organelles that carry out oxidation reaction that lead to the production of peroxides. Detoxification of various toxic compounds that enter the bloodstream is also done by peroxisomes. These use hydrogen peroxide generated during oxidation to detoxify the toxic compounds called peroxidation. Peroxisomes in the liver help in detoxification of alcohol. They synthesize plasmalogens, a component of the cell membrane of such tissues as the brain and the heart. Peroxisomes are the sites for beta oxidation of fatty acids. In plants, peroxisomes play very important role in seeds. Important function served by peroxisomes is plant cells in that during Calvin cycle, they helps to recycle phosphoglycolate, a byproduct of Calvin cycle. This is very important function as in the recycling didn't happen, a lot of the cell's energy and carbon would be lost. Spero zones. These are vesicles of 0.5 to 1 micron in diameter and single membrane body. Contains 98% of lipid, contains some acid phosphatase, limited to plant cells only and absent in animal cells in prokaryotic cells. Glyoxysomes. These are specialized peroxisomes found in plants, particularly in the fat storage tissues of germinating seeds and also in the filamentous fungi. In glyoxysomes, the fatty acids are hydrolyzed to acetyl-CoA by peroxisomal beta oxidation enzymes. Glyoxysomes possess additionally key enzymes of glyoxylate cycle, that is isocitrate lyase and malate synthase, which accomplish the glyoxylate cycle bypass. Glyoxysomes also participate in photorespiration and nitrogen metabolism in root nodules. Coming to cell division. The cell cycle is sequence of events or changes that occur between the formation of cell and its division into daughter cells. It has a non-dividing growing phase called interphase and dividing phase called mitotic or M phase. Here you can clearly see the 75% of area is contributed for interphase and only 25% is contributed for M phase. That means majority of the functions are completed by a cell in interphase rather than M phase. But M phase is very essential. How is it? We'll see here. Interphase. Inter means between, phase means aspect. It's a long, metabolically active phase between two successive mitotic cell division. It has three substages, G1, S and G2 phase. G1 phase is post-mitotic phase. The cell separates for DNA, RNA and protein synthesis. S phase is a synthetic phase. The duplication or replication of DNA and centriole takes place here. G2 phase is post-mitotic phase. Here, synthesis of proteins required for synthesis of spindle fibers. M phase, mitotic phase, is short phase, includes two processes like karyokinesis, that is division of nucleus, and cytokinesis, that is division of cytoplasm, resulting into two daughter cells. After M phase, the cell may enter either interphase to repeat the cell cycle or G0 phase to arrest cell cycle. Then the cells in G0 phase may grow and differentiate into different cell types to perform different functions. In the tissue cultures, three functions are very important like differentiation, de-differentiation and redifferentiation. All the three processes are associated and accelerate between G0 and G1 phases. Mitosis or the M phase is further divided into mitosis and meiosis. Mitosis was the means mitos means thread and osis means stage. Walter Fleming in 1882 studied mitotic cell division in animal cells and coined the term mitosis. This is a type of division in which parental cell produce two similar daughter cells that resemble the parental cell in terms of chromosomal number. It is also called equational division or homotypic cell division. Maintains constant number of chromosomes in each cell of successive generation. It occurs in somatic cells of the body, so it is also called somatic cell division. Mitosis occurs in two stages, one is karyokinesis and another is cytokinesis. Significance of mitosis. It maintains genetic stability within the population of cells derived from same parental cell, helps in growth and tissue repair, helps in the replacement of dead and worn out cells, means of reproduction in lower organisms. Metaphase. Esters are not found in plant cell as they do not have centrioles, hence called anestal cell division. Metaphase. In mitosis, meta means after, phase is appearance. Here spindle fibers are completely formed. The chromosome becomes short and thick with two distinct chromatids each. All the chromosomes move towards the center of the cell and arrange in the equatorial plane right angle to the position of esters to form metacentric or metaphasic plate. Chromosomes are scattered to spindle fiber at their centromeres. Anaphase. The centromere of all the chromosomes undergo longitudinal splitting and the chromatids of each chromosome separate to form daughter chromosomes. These move towards the opposite pole, forming equator by the activity of spindle fibers. Telophase. The daughter chromosomes reach the opposite poles. Chromosomes undergo despiralization to form long, thin thread-like structure called chromatin. Nucleolus and nuclear membrane reappears. This spindle fiber disappears. Cytokinesis. Cyto means cell, kinesis is movement. It is the division of cytoplasm. Cleavage furrow develop in the middle of the cell in centripetal direction due to the contraction of microtubules. It occurs till the edge of the plasma membrane meets. They fuse to form a separate membrane. In plant cells, cytokinesis occurs due to the formation of phragmoplast in centrifugal direction. The phragmoplast is formed by Golgi complex, endoplasmic reticulum, and pectin containing vacuoles. Thank you. I hope you liked my video. Do not forget to subscribe.